Okay, thank you. Thank you very much. My name is Alejandra Sierra, and I'm from the Virtanen Institute from the University of Eastern Finland. And during these 15 minutes of FAME, so I'm going to show you the different topics that we work in my group. So my group is a multidisciplinary team. That is, there are people that they have background in physics, in biology, computer science, and in mathematics. And all of us, we have a common motivation, that is to study the brain through imaging. But I would like to explain to you a little bit why we are motivating, what is our main focus of the research, and is MRI. So MRI is a non-invasive technique, and it has been extensively used in clinical settings already for the detection and the diagnosis of brain diseases and disorders. But still, if we think about the brain and MRI, so the information, even though is very useful, non-invasive, is very ambiguous when it comes to tissue microstructure and cellular components of this tissue. So one of our main focus of research is how to establish the contribution of the cellular components and the cellular composition to the MRI signal. So what I'm going to show you is a little bit this multidisciplinary approach and how we use all the different modalities to basically study this combination. So I'm going to start talking about a little bit data collection, because this is really crucial for us. So if we want to achieve our goal, we will need like different kind of imaging modalities, and this has to be collected in a certain way to be successful. For many years, we have been using animal models of disease. So we produce animal brain diseases, like, for example, epilepsy, traumatic brain injury, ischemia, Alzheimer, many others. We use rats and we use mice. And they give us one main advantage. It's like we can actually control how we produce the disease, and then we can image the animals in vivo doing longitudinal experiments. So we can actually image the animals in several time points. And then after the end time point, we can perfuse the animals, extract the brain, and do ex vivo high resolution imaging. And of course, the tissue is available for histology. So if you think about this pipeline, so this is very unique, because then so we can actually have MRI and histology from the same subjects. And then we can study our MRI signal and the one-to-one, the cellular components and the cellular composition directly to the MRI in the same subject. But now also we have developed the same pipeline for human studies. So we work with drug refractory epilepsy and also with tumors. So in this case, so we use this, we do patients that they come from Kuopio University Hospital. And then what we have is we have the opportunity to do preoperative MRI, and we do conventional and advanced sequences. I will show you something a little bit later. And then after resection of the tissue, because drug refractory epilepsy and tumors, the patients undergo surgery removing in the epileptic zone in the case of epilepsy and the tumor in patients. So we can actually get the tissue and be able to do ex vivo MRI and then histology. And again, so this is actually the perfect pipeline for us to get the information throughout all the scales. A little bit about the epilepsy project. So it's a project that is funded by Janne and Aatos Erkko Foundation, and it's a collaboration between 4 groups from University of Eastern Finland and Kuopio University Hospital. It's not only imaging, so here we have many other techniques like molecular and cell signatures, also neuronal function and data integration. And our motivation is to study how the tissue became epileptogenic. So just to show you a couple of examples of the preoperative that we do in the patients. So as I mentioned, we do conventional MRI, so basically basic relaxometry techniques, T1. So we do also functional MRI, so including MREC and resting state fMRI. And in structural MRI, so we do RAF, that is a method that is very sensitive to the myelin content, and also multidimensional MRI that I will explain a little bit later, but is sensitive to tissue microstructure. Just to show you some examples of the ex vivo from tissue resected from epilepsy patients, here you can see an hippocampus. This is a gyrus from the cortex of one of the patients. And here you can see high resolution T1 and diffusion tensor imaging in 3 different maps. And here you can actually appreciate the high resolution. We can see the lamination of the hippocampus very clearly, and also the lamination white matter, gray matter in the cortex. And from that, so we can get the histology. So here are examples of the non-sclerotic hippocampus, and here a sclerotic one. And here you can see examples of myelin staining knee cell for cell bodies. And here you can actually see the immunohistochemistry against antibodies that they have, like for nuclei in blue, astrocytes in green, and neurons in red. But we are not only interested in MRI in the conventional form. We are interested to go beyond the conventional MRI. We are interested in testing new techniques that they can be more sensitive to detect subtle changes in brain diseases and disorders. So we started many years ago working extensively with diffusion MRI because it's sensitive to the water molecules, how they move, and this basically reflects information of the tissue microstructure. So conventional techniques, actually, they give us one value that per voxel. So basically average all the information that voxel that is so complex actually get. And we can get interesting values, like, for example, anisotropy, that is organization, orientation, and also restriction. There are also other state-of-the-art methods that we apply that, for example, multi-fiber models that they can actually give us information about the organization, orientation, dispersion. But lately, so we have been implementing what we call multidimensional MRI. That is a technique that they give us not one value, not few values, but thousands of values per voxel. And then we can extract different kind of parameters related to the size, or the shape, or also orientation, restriction, and we can actually get relaxometry in the same acquisition. So basically the chemical composition of the tissue. And if you actually can compare, so the multidimensional is actually closer to the ground truth information. So of course, comparing the multidimensional with the histology, it will be basically a step forward for us for detecting different kind of cellular pathology in diseases and disorders in the brain. Just coming back to the hippocampus that I showed you before from one of the epilepsy patients. So this is the multidimensional maps that we can actually extract. Here you can see different contrasts related to the size and the shapes, the relaxometry, restriction, and also this rich information for voxel, it can be actually split. And we can get information from, for example, just white matter, gray matter, or free water. And having like thousands of points per voxel, we can actually represent distributions. So we are not restricted to represent the average of the voxel in a map. So these distributions, actually, they are quite powerful because they can actually give us information of differences that we can actually miss with the average. Like, for example, here, we can actually differentiate already between non-sclerotic and sclerotic hippocampi in the human brain. And going beyond this classification of white matter, gray matter, and free water, that is very rough estimation. So with multidimensional, what we can actually do is actually do data-driven classification. So we can classify all these thousands of voxels and be more precise, trying to give information of distinct cellular compartments. And this is basically our next step, try to validate what these different distributions can tell us from a specific cellular components. If we go to the other side of the story, so we need to validate or characterize the MRI signal. And for that, we need histology and microscopy. So for many years, we have been actually using light microscopy. So we do multiple stainings. So we basically section our brain. We do multiple stainings. And we do also 3D confocal. We do many other different techniques, not only these ones, but the routine ones is basically light microscopy. In our routine also is 3D electron microscopy. You see here an example of the hippocampus, and I will show you some examples also of the white matter later. But it's not only the imaging, so we need to do data analysis. And we have been over the years developing how we actually get metrics from the tissue. We have done traditional optical density analysis and many other kind of traditional quantification. We have developed advanced mathematical analysis that we can actually get parameters that they are equivalent. So what we get in MRI, like for example, a structure tensor or Fourier analysis. But now we are going to analysis for the whole brain, and we do tissue classification using autoencoders, for example. And this actually, it will give us much more power to analyze even the whole brain and correlate that to MRI data. In electron microscopy, so we have actually developed different kind of pipelines for imaging different resolutions at the same time. And also we have developed segmentation pipelines, as you can see here in the white matter. So these are basically realistic tissue models that they can actually be used not just for validation, but for very crucial biological information that we can extract with this information. So here you can see actually the segmentation of all the axons in the corpus callosum in the rat brain. But having multi-resolution information, we have developed also super resolution techniques with this data. So this is actually advancing a little bit. We can actually balance how much we can actually push the resolution versus the time of the imaging acquisitions. And then I will like just at the end to show you a little bit about what we can do for MRI-based prediction of the tissue microstructure. If we can actually predict different kind of information from the tissue from MRI. So what we basically do is basically whole section quantification using, in this case, segment anything. Then we create cell maps and we do registration between MRI and histology. And then we use regression analysis using random forest approach. And here you can compare maps of cell density into different samples. This is the ground truth. And then we compare with conventional MRI and different modalities, so the per voxel means and being resolved in multidimensional MRI. And this is examples of the area. And as you can see, the being resolved predictions are much closer to the ground truth than conventional or average in the voxel. So this is actually a very interesting future for us. So just to summarize, so we actually are a multidisciplinary group that we do different kind of data acquisition for the brain. We use advanced MRI and advanced microscopy, but other imaging modalities are welcome, the same as advanced imaging data analysis. So we are very happy if you have other ideas or you would like to collaborate in this regard. And just to summarize, so a multidisciplinary approach offers novel avenues for not just for validating MRI, but also for deepen our understanding of the healthy and the diseased brain. Thank you very much for your attention. And thank you very much for FAME and also for DREAM. Thank you.